Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. So, this is lecture 35, where I will discuss about the recent discovery of colchicine biosynthetic pathway. So, colchicine is basically an alkaloid, is amine alkaloid. It is produced by the plant called Gloriosa superba. SUPERBA superba, and another species colchicum autumnal, I put colchicum species, which basically produces this alkaloid. Now, why should we study colchicine? Colchicine has several applications including pharmaceutical applications. So, the most important one, it is used as a drug for treatment of goiter, that is a severe form of arthritis, and not only that, colchicine is basically used for cytological studies in order to make polyploid. In order to induce polyploidy, colchicine is used. So, colchicine plays an important role for not only for the pharmaceutical application, but also to decipher the fundamental plant biology. Okay. Now, colchicine, the structure of colchicine is pretty complex, and only the enantioselective selective synthesis. So, I put a color, enantioselective synthesis was only achieved in the year of 2017, but it was so complex that it cannot be applicable at industrial scale production. It cannot be applicable to produce at industrial scale. Therefore, the demand for colchicine to be made from natural resources available, and that sometimes makes the supply chain unreliable. So, therefore, there is a need for understanding the colchicine biosynthesis, so that later the molecular tools can be applied, leading to more production of colchicine in colchicum autumnal or in gloriosa or in some model plants. Also, maybe in future it is possible to produce colchicine de novo in another eukaryotic system such as yeast. So, now what is the status of biosynthesis? Okay. So, in order to study the biosynthesis, one needs to know or should have the idea of a precursor molecule. Fortunately, for colchicine, such molecule was indicated by the organic chemist. So, scientists have chosen the precursor molecule or the start molecule as 1-phenethyl isoquinoline, 1 phenethyl isoquinoline. So, this was considered to be the important precursor for the formation of colchicine. Now, how this 1 phenethyl isoquinoline to be produced in plants, that is again another question. So, we will address that a bit later. Let us go to the next slide. So, next slide is that the colchicine biosynthesis. So, this has been done by by the use of by combination of knowledges, a combination of transcriptomics, metabolic logic, and pathway reconstitution. So, these are the important steps to be considered in order to understand the colchicine biosynthesis. Okay. Now, when we talk about the transcriptomics, what they have done, that one thing was clear to them, that colchicine accumulates in the rhizome, that is the underground part. Sorry, rhizome. It is the underground part of Gloriosa superba. So, what they have done? They have done a comparative transcriptomic study. TRNSCRIP transcriptomics of leaf, stem, rhizome, okay. And then what they have done, from there they try to study the differential gene expression. We try to understand the differential design, and this transcriptomics was de novo. That is, no information available earlier about the transcription profile of this plant. So, from there they could get an idea that what could be the candidate genes for colchicine biosynthesis. So, that means the gene expression analysis will tell you, particularly as I said that as the rhizome is the part. So, definitely when you analyze the differential gene expression in rhizome, you will be able to find several transcripts which are not present in the other part. Now, these transcripts is properly analyzed, from there an idea can be generated that what will be the potential genes or enzymes responsible for the pathway. So, actually that was the starting point, and then once they come up with the idea that this could be the enzymes, and then they basically work out the chemistry, putting the putative substrate and putting the enzyme, and there what could be the product formation. So, again for example, I have discussed long back. So, as I have said, the substrate more or less is known from the organic chemist, from the study made by the organic chemist. So, and say you have discovered the enzyme Z, enzyme Y, enzyme X. That means the subsequent the genes are gene X gene, sorry. So, the X gene will encode enzyme X, maybe Y gene encode enzyme Y, and Z gene encode enzyme gene. So, then once they understand this one, that is what are the putative candidate genes X, Y, Z, and then what enzyme they will encode, that is X, Y, Z enzyme. And then if such enzyme is there, say methyl transferase, you know a substrate, then if a methyl transferase work, then it will make a potential product, so which could be B. Okay. Then there may be a hydroxylase, for example, which may be involvement of cytochrome P450. So, the product will be some sort of hydroxylated product. So, that will be C, and similarly D. Like that way, they theoretically created the concept, and once they created the concept, then they characterize these genes, they clone it, and they studied the functional expression of these genes individually in an eukaryotic system to confirm that indeed gene X converts A to B, say gene Y converts B to C, and then gene Z converts C to D. So, based on all this knowledge, what they have come up is basically the pathway which I am going to put it here. So, one phenethyl isoquinoline, as I said, that is the starting point. So, let us put that 1, that is the part of, I should what will be the heading? The heading will be part of the biosynthetic pathway of colchicine. So, 1 phenethyl isoquinoline QUI. Okay, the board is a bit disturbing. Q U QUINOLINE. So, this will be converted into several step reactions, and finally, it will produce a compound which is called S autumnal. So, S AUTUMNALINE. S autumnaline. So, Colchicum autumnale is the species, from there perhaps the name given S autumnaline. Now, this involves 6 steps, and including 4 methyl transferases and 1 P450 enzyme. And in the next step, this by the action of one unusual enzyme or one unusual reaction, that is 1 P450, that job was to do the phenol coupling, PHENOL phenol COUPLING coupling. It makes the product which is called androcymbin, isoandrocymbin. Now, isoandrocymbin subsequently by a two-step reaction where it is basically two steps. So, one is basically 1 methyl transferase, another is 1 P450, and that this is called basically the ring expansion. And this leads to the formation of N-formyl D-demecolcin, all Greek names, N-formyl demecolcin. So, it was subsequently predicted that N-formyl demecolcin will be converted into colchicine through subsequent steps, which may involve demethylase, deformylase. Okay, I think deformylase we should write before, which may involve deformylase, then demethylase and N-acetyl transferase. Leading to the formation of our target molecule, which is colchicine. Now, this part is still not confirmed at the molecular level, yet to be confirmed, but up to N-formyl demecolcin this is confirmed. So, that means up to this much from here, this is called the near completion step of colchicine synthesis. So, this paper I will discuss again the steps a little bit more detail. This paper published in the famous journal Nature in the year of 2020, volume 584, page number 148 to 153, and the first author is Nett et al. N-E-T-T, Ryan S. Nett et al. And this was published from Stanford University. And this paper is so impactful, the editor asked another scientist with a similar expertise to write a news and views article. So, news and views article also published in the same issue of Nature 2020, volume 584, page number 49 to 50. So, and they have given a nice name for this news and views article, that is the chemical know-how of the of the flame lily. So, to sum up what I have said, that the starting point for Nate and his colleagues is basically based on a proposed biosynthetic pathway given by organic chemists several years back. So, there what they considered that 1-phenethyl isoquinoline is probably the starting point of colchicine biosynthesis, and this 1-phenethyl isoquinoline is probably derived from amino acid phenylalanine and tyrosine. And this intermediate, that is 1-phenethyl isoquinoline, this to be converted to N-formyl demecolcine in 9-step reaction, the first intermediate that contains the molecular scaffold of colchicine. So, this conversion basically involves two unusual biochemical reactions. So, number one is a rare transformation which is known as phenol ring coupling. So, which is this one, and an unprecedented reaction that leads to the expansion of the molecular ring. So, that is somewhat unusual reaction which was not reported in the plant system before. So, that means phenethyl isoquinoline is initially made from amino acids phenylalanine and tyrosine. It is then converted into S-autumnalin in 6 steps by 4-methyl transferase and 1 cytochrome P450 enzyme. Then 2 other P450 catalyzes the unusual biochemical reaction. First one is a phenol coupling, and the second one phenol coupling, and as a result of this first one reaction it produces isoandrosymbin, and then a ring expansion which produces N-formyl demecolcine. Now, a 5th methyl transferase is also needed to convert isoandrosymbin into the substrate for ring expansion. So, the enzymes for the final 3 steps, in which the N-formyl demecolcine is thought to be converted into colchicine, is not yet confirmed at the molecular level, but it was predicted that deformylase, demethylase and N-acetyl transferase will do the job. So, okay. Now, this is in brief what has been elucidated by Nate and colleagues, and this paper published in Nature, what I said. Now, let us go to the some of the early steps of the pathway. Now, if I put this in a sequence, I will not write the name of the each and every substances, because it will be rather pretty heavy. So, to start with, say what I said, that the 1-phenethyl isoquinoline. So, 1 phenethyl isoquinoline. So, this is considered as a 1, that makes 2, 2 makes 3, 3 makes 4, 4 makes 5, 5 makes 6, then 6 makes 7, 7 makes 8, 8 makes 9, 9 makes 10. So, the enzymes are OMT1, then NMT, then CYP 75A 109, that is cytochrome P450, then again a OMT 2, then again a CYP 75A 109, then OMT, then again CYP 75A 110, then OMT 4, and then again CYP 71 FB 1. So, these all constitutes the pathway. So, the last one is the in formyl colchicine. The penultimate step was O methyl androthymine, and that previous one is number 8 was isoandrothymine, and then the 7 was autumnalin, and then subsequently previous steps. So, this is all the steps. So, these are all characterized. Now, now once they have characterized this, the next point was basically they want to check the biochemical function of all these candidate genes. So, they have used they confirm the biochemical function of the enzymes encoded, a function of the enzymes encoded by the 9 candidate genes. So, here they have used the model plant, which is a relative of the tobacco, Nicotiana benthamiana. So, what they have done? They have transferred all 9 genes by agrofiltration. Along with that, they have done feeding of substrate, which is the one which is 1 phenethyl isoquinoline, and then what they have done. So, then they they check for the production of this product, which is the N formyl colchicine, and all genes worked well, and as a result of that what they found that there will be the they achieved total biosynthesis of. So, what I put it in another color upon transient expression in Nicotiana benthamiana leaf, they found total biosynthesis of N formyl demecolcin. So, that means successful reconstitution of colchicine biosynthetic pathway in Nicotiana benthamiana. Now, they are not happy with this, they wanted to also express the early genes of the pathway that leading to the formation of the first product, that is 1-phenyl isoquinone. So, what they have done in this case, they have used 3 different modules. So, in the next one, metabolic engineering of colchicine pathway in Nicotiana benthamiana. So, what they have done, they also inserted the primary means they aim to achieve that the primary metabolic products will be utilized directly by the plant, so that it produces the substrate which is the starting point for the colchicine pathway. So, they have used 3 module. So, in the module 1, where they have done, so they have taken several enzymes from different sources, including Gloriosa superba, which starting with DAHP synthase, that is the starting point of this secumate pathway, and eventually it makes phenylalanine, and this subsequently by the action of multiple enzymes, for example, GS-PAL, GS4CL, GS-AER, GS-C4H, GS CCR, that leads to the formation of 4-HDCA. So, 4-HDCA, and here one thing I would like to tell, that is this okay, I will tell later. So, this is module 1, Now, they have also done the module 2, where L-tyrosine was taken and GS TYRDC or DDC. They have taken another plant from another source BV CYP 7685. This is from Barbarian vulgaris, and that ultimately makes dopamine. 4-HDCS stands for 4-hydroxy dihydro cinnamaldehyde. That is why the CCR was used. Okay. And AER stands for alkenyl reductase. So, now the module 2 completed. So, this and this will join together by the action of CJ NCS ocochlorine synthase, and that leads to the formation of 1-phenethyl isoquinoline. So, this subsequently module 3, which involves so many enzymes which I have already listed before. So, I am not putting all the enzymes. So, 9 and eventually it makes N formyl deme colchicine, and this subsequently by 3 successive reaction eventually will produce colchicine. So, what I said that these 3 steps are not characterized yet, but the penultimate product of colchicine has been achieved. So, that means they characterize the 9 genes from Gloriosa superba using transcriptomics, metabolic logic, as well as the chemical as well as the pathway reconstitution. So, pathway reconstitution they have done in Nicotiana benthamiana, where starting from 1-phenethyl isoquinoline until the formation of formyl demecolchicine. And what I said, formyl demecolchicine will be converted into colchicine by 3 steps, which have not yet characterized at the molecular level now, but it is confirmed that this is the near completion of the pathway. So, as I said, the genes were characterized from Gloriosa superba and functionally expressed individually in the another heterologous system, then they put all the genes together and they reconstituted the pathway in Nicotiana, and then what they did, they also reconstitute the whole pathway starting from the primary metabolic products, that is glycerol triphosphate and erythrophosphate phosphate, leading to the formation of DHP by putting DHP synthase from Gloriosa superba, and subsequently the genes for this secumate pathway leading to formation of phenylalanine, and then several early genes of phenylpropanoid including PAL, 4CL, CCR, C4H and AER, that ultimately led to the formation of 4-HDCA, that is 4-hydroxy dihydrocinnamaldehyde, and this joins with dopamine. Now, dopamine also they produce through module 2 by putting the genes of dopamine formation hiring from Gloriosa as well as from Barbaris, that produces dopamine. Now, this dopamine and HDCA joins by norcocloudin synthase, and which they have hired from CJ means Coptis japonica, and it makes 1-phenylethyl isoquinoline and subsequently all 9 genes inserted, and that makes the N-formyldemecolcin. That means, starting from the primary metabolism the end product achieved. So, this is really a wonderful work, and that is why this paper published in Nature. So, we have to wait for few more years when 3 penultimate and ultimate enzymes will be characterized. So, that whole colchicine can be produced, and that time maybe de novo colchicine in yeast, such interesting papers will come out in years to come. With this, I end this class. Thank you.